I'm Max Brown. I'm a genomicist, a bioinformatician at the Wellcome Sanger Institute. And a bioinformatician is someone who uses big computers to work on DNA sequence data. We have been working to understand apple DNA and apple genomes, where the genome is the complete DNA in a cell. And also, it's great to hear that some of these apples in this very orchard have been turned into cider. These leaves are crushed, the cells are broken apart, and the DNA inside those cells is extracted, dissolving the DNA into solution. When the sample has been sequenced, it comes off the machines in huge text files. The bioinformaticians turn all of this text, which is composed of A's, T's, C's and G's, and they assemble it into a long sequence. And that sequence then represents the genome of the apple tree. We have sequenced the whole genome of one apple cultivar called Brown Snout. It's this cultivar from Herefordshire from the 19th century. The genome of this is around 650 million base pairs long. So that may sound like a huge amount, but it's fairly small to medium-sized in terms of plant genome sizes. The other cultivar that we've been working on is called Kingston Black, which is a slightly older cultivar from Somerset. With these two cider apple sequences, we hope to understand it within the context of a wide variety of apples. So we have sequenced around 50 different cultivars, and in these differences we're able to say some really quite profound and interesting things about apples, such as how they're related to each other, whether they contain genes for certain traits like disease resistance, and even work out the evolution and the history of the apples. There are diseases called scab, which affect apples mainly in the winter. It's sort of a fungal disease which can cause huge losses, and some apple cultivars are really resistant to this. And it may be that the cider apples that we've sequenced don't have these disease resistances, but some of the other apples we've sequenced do. And so in future breeding programs, we can incorporate these into the next generation of apple breeding. Modern orchards have trees that are planted very close together and are in a pretty different circumstance to these trees of old. So perhaps we want to save even more space, but maintain our yields. We may find genes which underlie how early a tree will flower and produce crops, and also its tolerance to shade and competition. And this will allow future crop breeders to really get the best yields out of their crops.